Thank you, Divya. And first of all, I would like to thank Gnanam ma'am for inviting me for this to give this talk. It is always there in my mind. I have to do something for my for the university that has taught me the basics, and I think this is a great opportunity to get started doing something back to what I have learned. So I would like to thank Gnanam ma'am for this invitation, for the chance to share my knowledge, and I'm very happy to present you pretty much what I did during my PhD for the last 4 years here in the Wageningen University. So, good morning, everyone, for who is attending from abroad, and good afternoon, everyone who is attending from India. I will be giving a talk today on the invisible evolution, lessons from the deep past. Actually, there is a reason why I chose this title, because this is also for the students of bioinformatics or biotechnology from the university, and I thought I would put a bit of emphasis on my journey as well, so that it would give you a insight about how I have been traveling through from my bachelor's till the point where I am now. So, as I said, I'll be talking evolution, but there are two sides of it in my talk. So, one is the visible part, and the other one is the invisible part. So, for the visible part, one thing is it's a very recent past. So, I'll be talking about my own evolution, how I have traveled from all my schooling till the point where I am, and what the things that I have come through. And the other one is the invisible evolution, where I'll be talking about the 4 years of my PhD thesis. So my evolution is very, very simple, but I'll be talking mainly about the PhD thesis, where we have studied the deep evolution going back through a billion years. So to give you an overview of who I am and where I'm from, so I'm born and brought up in a small coastal village in Andhra Pradesh, where I did all my schooling, and then I've moved to Coimbatore, Tamil Nadu Agricultural University in India, where I did my bachelor's in bioinformatics, and I've passed out in 2011, almost a decade. And then briefly, I worked at IRI for a couple of years, International Rice Research Institute, working on understanding the genes involved in the drought responses, or in general, any stress response systems, by studying the quantitative trait loci needed for a particular, that are responsible for a particular trait. And then after working there for a couple of years, I've moved to Wageningen University in Netherlands, where I am now. So I did my master's, and then also I did my PhD, and I'm continuing as a postdoc now here at Wageningen. So yeah, I think pretty much this was the reason that I was able to learn so many things, because that's the main advantage that I had being a bioinformatician, and also by traveling to different places, by interacting with different people. A lot of strengths were there for me throughout my journey. I think this is pretty much the reason that I've been able to assimilate or cooperate different things, because I had so many different projects, I had different mentors, and I have worked in different places. So I was able to taste different cultures. I've gained a lot of experiences, a lot of interactions at a personal level with different people, and more importantly, I gained some patience throughout this travel. I think from a personal point of view, this has been a very advantageous journey, very good journey so far. As well as being a bioinformatician, I had a lot of perks also, because bioinformatics, I strongly believe that the main core of bioinformatics is about how good you deal with data and algorithms, and whether you have a good biological question to answer those questions, to use the data to answer the questions that you are interested in. Throughout this process, I think the big advantage for me, being from the professional side, was that the data is purely open source these days. So the amount of information that I got from the public data sources is so good. The number of platforms that we could use nowadays with the advancement in technology has been, I think this is just the right time. It's always a right time, because the technology has been progressing so rapidly, that you always have a new data, new algorithms to deal that big data, and you can always make these nice collaborations to ask different biological questions using different platforms. I think that has been a very good step so far. And I would like to put an emphasis for a main aspect here, that is the open source. I think many of us, especially being from different labs in a developing country, we always had problems that we can't spend so much money for generating new data, or we don't have such resources to deal the data as well. I think this is pretty much the advantage I say nowadays, that the data is pretty much open for everyone. So whoever is working, sorry, question. Okay, so pretty much whoever is working with a data nowadays, they put the data online. Whenever there is a whenever there is a publication, people submit the data out, and then at the end, we all have access to the data, and that's why I put pretty much emphasis on the open source. And I think that's the reason I was able to complete my PhD very successfully, because all of my PhD, the main source of my PhD data is open source. And that is what I have done in the last 4 years. I have utilized this data to understand the evolution of auxin response system in plants. Uh, uh, I think I would say strongly say that that has been the strength. But you don't worry about the big words that you see on the screen. That is about the evolution, about the of a billion years of auxin biology. I'll briefly take you through different steps I'll briefly explain you the different words that I'm talking about. So I think we need to understand the basics of evolution. If you want to understand the basics of the plant evolution per se, we need to understand the evolution of life in general. So when the Earth formed around 4.5 billion years ago, there was no life on Earth. It was kind of a non-existent matter. Then all of a sudden, not all of a sudden, at certain point after 1.5 billion years of evolution, slowly the life started to form on the Earth. And when the life has started, it was all in a small, simple forms. At certain point, the simple forms or the single cellular organisms have started to grow in a complexity, and it has given rise to different systems. For example, as you see, the earlier plants were nothing but the hydra and all other small unicellular organisms that float in the water. But slowly, the life has transitioned from water, that is somewhere from the fishes, and then to the land reptiles, and then to the mammals. So there has been a stepwise evolution throughout life, and the evolution is a never-ending process. It has been going on, and it is still going on. And as you clearly see, it takes a lot of time to evolve from one species to the another. But throughout the process, there has always been certain gains going on, and there has been a gradual increase in the complexity of life. In the same way, in plants also the same thing happened. So the initial stages of life were the small green algal systems that were there that were living in the water, mostly the marine waters, and then the slowly life transitioned into the fresh water. So the earlier stages of life was in the water. Then slowly the life has moved to the edges of the ponds or the moist environments on the soil, where the plants started to grow horizontally on the surface, not vertically, horizontally on the surface. But when the plants move from water to land, they need to develop more advanced systems because the water is scarce. They have to build a root system with a vascular system to suck up all the water and to transport all the nutrients. And then further, the plant started forming seeds, and then finally the flowers. So whatever we see on Earth today, the majority of the plants have not existed quite a long time before. It has been a stepwise evolution of small innovations that has happened through the last billion years easily. So as you clearly see, there has to be a stepwise evolution, and there has been a stepwise evolution, and that is what we are trying to uncover now in different aspects. And the aspect that we are working on here in our lab is hormones, especially the plant hormones, auxin. So hormones are very important for growth and development. For example, in human beings, there are different hormones like thyroid, melatonin, insulin, testosterone, and progesterone. For example, if insulin is not produced properly or not metabolized properly in a human system, it leads to diabetes. And in the same way, for example, testosterone and progesterone, if they don't work properly, the reproductive systems do not function properly in a human body. So there are different hormones that does different functions, but they are all coordinated in certain way to keep the whole human body up and running. In the same way, in plants, there are different hormones like zebrelin, strigolactone, cytokinins, abscisic acid, jasmonate, and then auxin. So among all these different hormones, as I said, we are interested in auxin, one of the key player in plant growth and development. Now that I mentioned the importance of hormones and especially the importance for growth and development and to maintain the healthy functioning of the plant. I'll tell you a bit more about the auxin hormone because that is the one that we have studied and we are interested in. Auxin is a generic name for a chemical molecule called indolestic acid. It is a small molecule, as you can see on the screen. It is produced in the plants. So if you go a bit deeper into the system, plant system, what you see is that you have multiple cells in a particular tissue, and each cell has different organelles. So every cell has these proteins, or you call it machineries, where a particular raw product or raw material is converted into a final product through these proteins. So in the case of auxin, the raw material or a starting point is the amino acid tryptophan. The tryptophan is an amino acid that is a kind of starting point for the auxin biosynthesis pathway. There are different routes of auxin biosynthesis, but the main route of auxin biosynthesis that is well known or well characterized, as well as the most appears to be the most abundant one, is the canonical TAA pathway. So TAA is an tryptophan amino transferase, and then there is a monooxygenase called yucca monooxygenase. So these are the two steps to convert tryptophan into the indolestic acid, that is the auxin. Once the auxin is produced in the cell, it has to be transported to wherever it is needed. So there are different proteins called the pin proteins. Transportation. Please mute your. Thank you. So there are these proteins called pin proteins that are needed to transport the auxin from the point of synthesis to the point of need. And then once this auxin is transported to the nucleus, the actual functional pathway starts. So the auxin is produced, transported, and it is finally is needed to send signals around. And that signaling pathway is called the nuclear auxin pathway. And the whole pathway is, as you get from the name, the whole pathway happens in the nucleus. So once auxin is transported into the nucleus, there needs to be a protein that senses the auxin or that binds to the auxin, that is called the TIR1. And then this particular protein, in the absence of auxin, these auxin proteins are the transcriptional co-repressors. So when there is no auxin in the plant, auxin proteins bind to the transcription factors called the auxin response factors and block the pathway. But when there is auxin, auxin acts as a glue, as a binding glue between these two proteins, the TIR1 and auxin, and leave the auxin response factors free so that they can do the job of transcription. So in the absence of auxin, there is no binding between these two proteins and the auxin response factors are free. So there is transcription, and in the absence of auxin, auxin proteins block the auxin response factors. So there is no transcription, let's say. So this whole pathway or this process is called the nuclear auxin pathway. And the question that we had was that out of billion years of evolution, out of the different proteins that we know are related to the auxin pathway, either for the production or the signaling, the question is how the complexity has evolved throughout the evolution. So the hypothesis that we had was that the organisms that were living in the water, or the species that were living in the water, are a simple single cellular organism. So they are more simple and more or less complexity is there. And as the evolution has progressed, the system has become more and more complex. So there are more components of this pathway taking have come into play to make the system more accessible for the need of the complex organism. So that was the hypothesis that we had. But uh, beyond this hypothesis, there are a couple of questions. The basic questions that we wanted to ask was that at certain point, these proteins has come together at certain point, and when the origin of these proteins has happened. That was the first question that we asked from the study. And then the second question is how the different components have evolved throughout the evolution from the water to the vascular system to the seeds and then finally to the flowers. So throughout this transition, we wanted to understand how the evolution has happened in this. But as I said, we are talking about a billion years of evolution, which means that there are thousands, hundreds of thousands of species that have come into existence, and many have been extinct, but there are so many species still that are so many species that are extinct, as well as there are many species that are living today throughout the billion years of evolution. But which means that we are talking about a few thousands of plants to study, which contains hundreds of thousands of genes. That's already almost more than trillion nucleotides. So to study all these information, we need a good data resource. And at the time I started my PhD, this 1000 plants or the 1 kp data was pretty much non-public, but now it is a public data. So we have utilized the power of this 1000 plant transcriptome data. So I reiterate, this is a transcriptome data, not a genome data. So you have all the expressed genes from small green algal species till the flowering plants. So yeah, this is a very good data resource if you want to study a global patterns of anything. In this case, we have studied the evolutionary patterns using this data. So this data is now public, and this contains more than 1,300 species of plant transcriptomes in every single stage of plant evolution. So we have utilized this data, and the first thing that I did was, because the data is so much and we are talking about so many genes and so many gene families, I thought the first step is to make a nice analysis pipeline. So what I did in this case was the first I developed a pipeline using already existing tools. So I didn't write any of my own tools, but I had to write some small scripts using Python and Shell to put all this information together. So, but the advantage is that, as I said, there are all the tools that we use in a common day purpose to dig the data or to mine the data. Of many of it are pretty much available. We just need to use it in the right direction. So, and that's what I did in my pipelines here, is that I've used, I'll briefly tell you what I did for this pipeline. So we have query sequences. So I take, for example, if I'm working with one gene family, I take the sequences from unknown species like Arabidopsis, the model organism, or rice, and then I use them as a query, and I dig the data. So among these 1,300 transcriptomes or some other proteomes, I do a blast search, and I get all the sequences, the homologous sequences. So that's the first step. I do a blast using a query sequence, and then get all the data out. That is all the homologous sequences. Then in the next step, what I did was that once you get the sequence, you need to identify what domains are there, whether the protein that you picked up has the domains that you're interested in or not. So whether you get a transcript sequence, then you convert it to a protein, or you directly get a protein sequence. And then I try to identify what domains are there, so that is a kind of first filter to say that, okay, this particular protein has these domains, so this is the one that I'm interested in. So with the ones that I'm interested in, I take them, I do a bidirectional blast. So which means that I take those proteins, blast them back to the query complete proteomes. So it is a kind of cross check whether in both the ways you're picking up the same protein or not, or whether the protein that you picked up belongs to the same family or not. So here I did a bidirectional blast. So the idea here is to filter the orthologs. So you get more closely related sequences in the third step. And then finally, once you do the alignment, you do your phylogenetic tree construction. So this is pretty much the brief overview that you do a blast, you do a domain identification, you do some filtering for your final set of sequences, and you go for alignment, and then finally you build a tree using a maximum likelihood. Here I have used different maximum likelihood algorithms to compare the trees that I get from different programs and to choose the best one finally that represents the biological reality. And once I have developed this pipeline, we tested on different gene families. We studied different gene families in the auxin pathway. I'll briefly reiterate. So there are two different gene families or enzymes or proteins that are involved in the synthesis of auxin from tryptophan. That is the TAA, the aminotransferase, and the yucca monooxygenase. So first we wanted to study the evolution of these two gene families. So using the pipeline, I get pretty much trees like this, which contains a few hundreds of taxa, if not thousands. Usually I start up with a bigger data set, few thousands of taxa, and then I trim the tree to a certain extent that the biology needs to be represented on the tree. But you don't have to worry about the details. I have a simple representation, but we did the same study for the yucca gene families also. But I would like to tell you one thing in this is that whatever the color you see represents to one clade of evolution, which means that, as I said, there are green algal species, the basal land plants, liverworts, hornworts, mosses, and there are lycophytes, ferns, and then finally the seed plants, that is the gymnosperms, and then the flowering plants, that is the angiosperms. So each group has been given depending on the species or the clade they belong to, they have been colored here. So if this particular group, for example, here, this is a green algal group, what you can say from this is that they are coming from a common ancestor. So which means that the green algae in its ancestral state has one copy. And if there are many species that are later there in the evolution, they may contain multiple gene duplications specific to that family or specific to that species. So here we are looking into the concept of ancestral genes. So in the same way, we have utilized this information to develop the schematic. So I'll tell you one example from this, so you know exactly what I'm talking about. For example, if you look into the TAA gene family, that is the first one in the pathway, what you clearly see is that I mentioned here that there is one copy of TAA proteins in the charophytes, that is the green algal species. And then during the land plant transition, it has been duplicated into two copies. And then that particular copy has been kept as a single ancestor. As I said, ancestral state is a minimal number of copies present in every organism in that particular lineage. So every liverwort must have one copy of the species that we have studied. So I think, as you can clearly see, there are two clades in the evolution. And for example, the liverworts are given in this purple color. So there is one clade of liverworts here. So that means that there is one ancestral copy here, and there is one ancestral copy here. So there are two ancestral copies, and that's what you see here in the liverworts, that there are two ancestral copies. And in the same way, we studied for all the clades in evolution for both TAA and yucca. I think the major message from this here is that there has been no duplications in the ancestral states in the TAA family since its origin in the land plants, since its first duplication in the land plants. But on the other hand, the second player in the pathway, that is the yucca, you clearly see that there has been a duplication and there has been losses. So there has been an independent loss. So the red dot here represents that mosses have lost this particular copy. And then there has been a continuous discontinuation once starting from the seed plants, that is the gymnosperms. But in the other clade, there are no losses. Instead, there are duplications. So as you clearly see, the vascular plants started to show a duplication in the gene families, further in the seed plants and further in the angiosperms, the flowering plants. So as you clearly see, the flowering plants have much more number of copies. Now this is about the auxin biosynthesis. Then we have moved to the next part in the study, that is the nuclear auxin pathway. once the auxin that is produced enters the nucleus, this pathway kicks in. As I mentioned before, the TIR1 is the receptor, auxin-A is the co-repressor of auxin response factors, and auxin response factors are the transcription factors that bind to DNA and does the transcription. So we have asked a bit more deeper question for this. So what we asked is when the particular proteins have been originated. So what exactly I mean here is that if you take one protein, that is the auxin response factors, this particular protein has so many different domains. So the basic idea here is that multiple domain containing proteins have different origin points. So it's not that the protein all of a sudden existed in one form. There has to be the different domains that have evolved at different stages. They came together at certain point or to form this canonical protein. And in the same way for auxin-AAs and TIR1 AFBs. So all these proteins are the multi-domain containing proteins. And interestingly, because as I said, the auxin proteins and OFF proteins interact, there has to be some domain that interacts. And this PB1 domain is the one that interacts between the, that is used for interaction between the OFF and auxin-AAs. So the first question that we ask here is that auxin response factors and auxin-AAs and TIR1s, how and when the different domains came together. And as you clearly see, so the carophytes are the green algal species, the chlorophytes are the primitive green algae, and the red algal species evolved before the green algal species. So without going into the details, what I would like to show here clearly is that the existence of a complete protein you clearly see only in the carophytes, which is the common ancestors of land plants. So before the green algal evolution or the streptophyte evolution, you see different domains, but they are not always together, or they are always mixed or combined with some other proteins. And at certain point, this particular protein has come into existence, and that has been maintained throughout in the evolution. So the idea here is that the carophytes have came into existence. I mean, these proteins have come into existence in the carophytes, which is further duplicated in the land plants. Like I showed for the TAA and UCA proteins for the auxin biosynthesis, we did the same analysis for the three gene families, that is the AFs, AUXIAs, and TIR1 AFBs. This is actually a pretty lot of information in this slide. But I would, to simplify that, what I would like you to focus on two things. One is, in some proteins, there is initial duplication in the carophytes. For example, in the auxin response,
Yeah. Maybe please connect your presentation from the previous slide so that people may be able to follow it. Thank you. Yeah.
So I'm sorry, there has been a connection dropout. So what we did in this point is, once we studied the different steps in the evolution, then we wanted to understand the complexity of the regulation. So because this is a transcriptional pathway, so the question is, having more number of copies means that there is more transcription or the more complexity in the transcription. So that's what we wanted to study. And for that, what we did is we picked up a couple of species from carophytes, one from the liverworts, hornworts, and then mosses, and then ferns, which has different number of copies in the system. So what we did is we picked up these species, we treated them with auxin, and we did a RNA-seq study. So we did an initial 1-hour treatment because we want to study the immediate auxin responses, the primary auxin responses. So I'll show you what we got in that study is that if you look into the carophytes, that is the green algal species, this was quite unexpected for us because, as you clearly see, there is hardly one gene or one transcription factor, that is the auxin response factor, related to the auxin pathway in these species. but you clearly see there are more than 1,000 genes that are being differentially expressed in the green algal species. That was quite unexpected for us, but it could be the reason that this amount of concentration of auxin is too much for this plant or for these species, that it might be a purely a stress response, or that is a something very different, or a nutritional response, because tryptophan is also a kind of nutritional molecule and utilized as a starting point in different pathways. So this is definitely not an auxin response. That's what we thought. But what we studied is that we looked into the different bryophytes. We looked into the different bryophytes here. So the idea here is that we studied how, based on the complexity, we checked if, as the number of copies increase, is there an increase in the transcriptional regulation. So, for example, there are three copies of auxin response factors, and we see a few amount of genes that are differentially expressed in Anthoceras. It is a hornwort, it is a basal bryophyte. And then there are 4 copies, and then we see a similar amount of transcriptional response. But moss, which has a recent whole genome duplication, we clearly see that there has been more than 1,000 genes being differentially expressed. But I think one thing that is common to all of these is that the downregulated genes, that are the ones that are given in the blue, has dominated than the upregulated genes. But here is the surprise, that we found completely opposite a pattern of interaction or the expression in the ferns. So what you clearly see here is that there is more upregulation than the downregulation, and there has been a switch here in the transcriptional regulation. And these are the vascular plants. As we saw before, there has been more duplications in the gene families, and there has been more complexity, as you clearly see from the differential expression patterns. But we wanted to ask one more question. If this is not a transcriptional response to auxin, we need to understand if there are any other common players or any other genes that we already know are involved in the transcriptional responses are given in these or identified in these pathways. For that, what we did is I took all the downregulated genes, I took all the upregulated genes, clubbed them together, and then I wanted to see what are the common genes in all those upregulated genes and all those downregulated genes. So for the initial studies, gene ontology was of no use, because at the time we were studying, for many of these species, there were no genomes were available. So we couldn't identify the genes, we couldn't do the gene ontology studies to find what these gene families are involved in. For that, what I did was I took all the differential expressed genes, I did a pairwise blast, so all against all sequences, which means that every closely related group has to blast one against each other, has to have a hit or a match against each other. And then that would give you a big table of pairwise matches, which means that so you have different queries sequences and use the same query sequences as the database as well. You do a blast, and then this whole bunch of data, you get a lot of data that you really...
Hello, Sumanth.
Yeah.
Can you please, there is some background noise actually. Can you please take care of that?
I'm not sure, actually. Sorry, it's pretty quiet.
That's okay.
This.
Okay. Yeah, please go on.
So,
Yeah.
So we get these big matches, or big, because there are thousands of genes. Once you start matching one against the other, you clearly see that there is a big table at the end. So the idea was that, okay, then I thought, okay, we need to visualize this data somehow to get this information in a understandable way. So for that, what I did is I made a network of blast hits. So if there are, for example, here, if there are two different proteins, and if there are two lines between them, which means that when you do a pairwise blast with each other, those proteins have been identified to be more closely connected. So in a pairwise blast, one against the other, you clearly see that they are closely related to each other as the top hits. So which means that the more closely connected a network is, which means it is a one family of proteins. And the more number of balls that you see, that means that the more number of gene families are present, or genes are present in that particular family. And I've used the functional annotation to annotate each of these clusters. And then what we did was that we picked up few of the clusters for further analysis to see if So the more number of colors, so each color here represents one species, and each dot represents one protein. So the more the number of cluster size is that more proteins are there for that gene family. And we did the same for the downregulated gene families, and we clearly see that there are some gene families that are pretty much conserved across different species, or to say the pretty much compared, pretty much commonly regulated across different species. But this is again purely a blast result. We wanted to confirm this, so we did some testing in the lab, and what we found out was that indeed, for example, you clearly see even with the qPCRs, we were able to confirm that some of the gene families that we picked up from the previous analysis were able to be commonly upregulated across different species or commonly downregulated across different species. So it was clear that there are, I think the main outcome of this is that there are some gene families that are commonly regulated throughout the evolution. And we also had to do some experimental validation to test these predictions. I won't be going into the details, but the major idea that we got here is that our previous collaborator, Hirota Kakato, he did a mutant analysis using CRISPR-Cas in one of the basal land plants, that is the Mercantia polymorpha. It's a liverbot. And what we clearly see is that we were able to test the hypothesis that some of the genes which were involved in the auxin response pathways are indeed regulated in a positive way as we expected, along with the differential expressed genes. But more information, we have put it all this in the eLife paper that we published a couple of years ago. But I think I gave a kind of idea of how I use this data to understand evolution. So the summary here is that there are different switches. So for example, in the green algal species, there are few switches. Assume that every protein is a switch. Depending on the necessity, it turns on or off. And then as the evolution has happened, the number of switches has increased. So when there is a new innovation, original switches has been retained and a new switch has been created or duplicated. And in the same way, at different stages in the evolution, different switches have been formed. And depending on the complexity and necessity, these systems develop or retain these copies or to maintain these complexity. Before ending my talk, I would like to give you two examples of what we did further in terms of evolution. So what you clearly see here is that in the previous study, for example, we used a multi-domain proteins. So it is very easy to study multi-domain proteins because you have so many anchoring points to understand the complexity or the structure of the gene from its starting point. But when you want to study a single domain, it's a bit difficult. So to test this method, we also picked up the PB1 domain. It's an oligomerization domain that is common to both off and auxiliary proteins. We studied that even beyond. So we studied it throughout the animal kingdom, plant kingdom, protozoans, diatoms, and fungi as well. And we also studied a bit of evolution of this to understand how the different gene families or different copies have evolved from a common ancestor of the eukaryotes. Because we studied so many of the eukaryotes, and then we wanted to study what is the starting point of evolution of all these eukaryotes. And then I clubbed this information with a random forest. So I took the sequences because we have sequence parameters, for example, the length of the protein, the amount of tryptophans, the molecular weight, the net charge. So I took all these parameters. We created a random forest model to test or to identify or characterize which protein belongs to which gene family, and we were able to do it with a pretty much low error rate. So the whole idea here is that we were able to combine the evolution part with the random forest and finally the modeling to understand how the structure of the protein has been different in the different gene families. Here it is shown for the plant kingdom, but as I said, I would briefly like you to show that we were able to test the data with a single domains, and you can find more information in the paper that we have published recently. And one more thing is we tested with multi-domain families, but from a different perspective. So because we also worked with plant cell polarity in our lab, so we identified a few years ago a protein called Sozeki, And we have studied in a similar way for the previous families. We studied how the evolution has happened in these gene families throughout the evolution. And one thing that I would like to point out here is that apart from in the plants, we don't know this domain architecture. So we identified using MEMI for motif prediction. We identified these unknown families. We characterized them. We studied the evolution even in fungi, metazoa, and also in the diatoms using various public data resources. So the method is not only applicable for one particular data set, it is also applicable for different data sets that are available. So this is a diatom data set. It is again a transcriptome data, and microcosm from JGI is a genome data. But the whole idea or the essence of this study is that we were able to develop some efficient pipelines that we could use it for the public data to understand the evolutionary patterns, and all the parts that were there in the green algal systems, and the complexity has evolved further in the land plants. So starting from ferns, there has been a stepwise increase in the number of copies, and that is what we studied and we saw. With that, all the information that we have studied now is a major core of my thesis, and I would like to point you. If you are interested, I would like to recommend you to look into my thesis, Evolutionary Analysis of a Billion Years of Auxin Biology. It is again open access thesis, which you can find under this particular link, DOI number. With that, I'd like to thank all the members of biochemistry department where I'm currently working in and where I did my PhD as well. And a special thanks to my mentor, Dolph Weyers, here in the right. And has been a very good support throughout the duration. And we were able to publish all this work in a nice journals, and we were able to come up with a nice hypothesis to do further research on auxin biology. With that, I would like to thank all the members of our lab, and a special thanks to Hiro Takakato. He was a previous postdoc in our lab, currently in Japan, who has been a big collaborator to do all the wet lab experiments in the studies that I have shown you, and one of the experts in Mercantia auxin biology. And with that, I would like to thank you all for your attention, and I'll be happy to take any questions.
Thank you, Sumanth, for the wonderful talk. Ma'am, do you have anything to say, ma'am?